So here we will talk about different mutations. Now these mutations are essentially the spelling mistakes in your genetic codon or in your genetic code, right? Grammatical errors, spelling mistakes, all those, you can call them these. So these are mutations. Now there are major 6 types. It could be silent, missense, nonsense, trinucleotide repeat expansion, splice site mutation, and frameshift mutation. First is silent mutation. Silent mutation means that there will be a mutation, but it won't, it won't alter, it would not cause problems, right? It won't change the amino acid sequence. How does this happen? We know, as I told you before, that the code is degenerate. There is code degeneracy, right? Because of code degeneracy, serine, this is serine, is coded by both UC and UCU. If this A gets mutated into uracil, it won't be a problem, but it will be a mutation. And this mutation will still code for serine. So the amino acid polypeptide, there will be no problem in that. There will be no problem. There will be no disorder. Everything will be in order, but there will be silent mutation. Such mutations are called as silent mutations. So silent mutation is also a point mutation, that only one base is getting mutated, right? But still it's not being a problem. But in some other cases, if there is a point mutation, ladies and gentlemen, then it might be a problem, like in sickle cell anemia. Sickle cell anemia is a point mutation, right? Okay. Then we have the missense mutation. In missense mutation, what happens is that it is missense. Another one amino acid is coded for another. So there is a replacement of the amino acid, right? So serine is coded by UCA and proline by CCA. If U is mutated right here for C, then this will code proline instead of serine. Now it's a problem. Now why a proline is inside? Now a proline is inside the polypeptide rather than a serine. Then we have the nonsense mutations. The nonsense mutation, you see nonsense, which means there is a stop codon. UCA is now UA. C is now mutated for A. Now this is stop codon. This will cause, because this is stop codon, it will cause a stop. It will stop the translation process, and this will cause the formation of a truncated protein or a short protein. Short. Definitely defective. Then we have the trinucleotide repeat expansion, which means that it is a lot of gibberish codons, right? There are a lot of gibberish codons, but they are gibberish, but they are not introns. They actually gets translated into a protein. So CAG in a mRNA code, CAG is going to repeat a lot, and this is gonna code for glutamine, and over again there is going to be glutamine, and this is going to produce long proteins, and they might be misfolded. That happens in Huntington's disease. Trinucleotide repeat expansion. Then we have the splice site mutations. In splice site mutations, we might have a problem in splicing, right? I have already done a video about splicing. So there might be either a repeat expansion, there will be a repeat expansion in splice site, and the splicing will be defective. When splicing is defective, then of course the proteins are produced that are problem. The dystrophin is like, for example, in myotonic dystrophy, dystrophin is defective, and that is because of splice site mutation. Splicing is altered. It could include introns, it could include faulty exons, half exons, or that's all that. Then we have the frame shift mutations. Now in frame shift mutations, I'm going to go over this in slight detail, not too much. Frame shift mutations, I'll tell you, like A, U, G, A, C, U, G, C, U, C, G, like that, right? Let's keep this one. So 5 prime to 3 prime. If, for example, this G is removed, let's remove this G. Now AUA, CUG, UCG, right? It is now going to work like that. See, this is a frame shift mutation. Now there is code is now read differently, right? What if there is an addition? If there is G, but now here I add C as well. Again, this is going to change. AUG, CAC, UGU, CG, and moreover. So you see, this is also addition and deletion of one nucleotide or one base is messing up the frame of reading. It is messing up the frame of reading. But if what I do instead is I actually remove this UCG, I remove this, right? I remove this. Now the frame of reading is going to be the same, but still there is a disease called as the cystic fibrosis, in which phenylalanine is deleted, the whole codon is deleted, and that causes cystic fibrosis, that causes problems with the CFTR protein, and that's that. There are frame shift mutation. Even though the reading of the frame is not getting disrupted, but still we we have this problem in cystic fibrosis. So that is frame shift mutations. Thank you so much. Goodbye.